Pyruvate. Pyruvate oxalostate to phosphoenol pyruvate is catalyzed by phosphoenol pyruvate kinase carboxykinase. Phosphoenol pyruvate then starts gluconeogenesis and synthesizes glucose. It is important to remember to form glucose from pyruvate through gluconeogenesis. Body needs two enzymes, pyruvate carboxylase with biotin and phosphoenol pyruvate carboxykinase. These two reactions are irreversible. It cannot be reversed by the same enzyme. Pyruvate forms malic acid. Pyruvate can be converted to malic acid through oxalostate in the mitochondria. Again, this is a two-step reaction. Pyruvate will first convert to oxalostate and then oxalostate will convert to malic acid. Pyruvate to oxalostate is catalyzed by pyruvate carboxylase and requires carbon dioxide. Oxalostate to malate is a reversible reaction by the same enzyme, malate dehydrogenase. The reaction requires NADH2. Malic acid may enter TC cycle or forms malate shuttle in the mitochondria. Pyruvate forms lactic acid in anaerobic glycolysis. In the presence of oxygen, pyruvate is converted to lactic acid, catalyzed by pyruvate dehydrogenase. This reaction occurs in the cytosol and is reversible. Reaction is important in RBC and WBC. We don't have mitochondria, hence they depend entirely on anaerobic glycolysis for energy. The reaction is also important in exercising muscles, which become hypoxic during strenuous exercise, resulting in increased production of lactic acid. Pyruvate can synthesize amino acid. Pyruvate can be converted to alanine. It is a reversible reaction catalyzed by SGPT-ALT with coenzyme pyridoxal phosphate. Pyruvate in bacteria forms alcohol. In bacteria, pyruvate can be converted to produce alcohol. Pyruvate to alcohol is also a two-step reaction. First, pyruvate is converted to acetylaldehyde with release of carbon dioxide, and then acetylaldehyde is reduced to ethanol, that is called alcohol. Coenzyme NADH2 is required to reduce acetylaldehyde. This reaction is responsible for fermentation of alcohol by yeast and some bacteria, including intestinal flora. Krebs cycle or citric acid cycle. Krebs cycle or citric acid cycle is not exclusively a carbohydrate metabolic process. It is the main or central cycle for the oxidation of carbohydrate, protein, and lipids to produce energy. It is an amphibolic process. Anabolism and catabolism is taking place simultaneously. It occurs in mitochondria in close proximity to respiratory chain. No ATP is utilized in this cycle. The cycle starts with acetyl-CoA and oxalostate. It produces 3 NADH2, 1 FADH2, 1 GTP. Complete oxidation of 1 acetyl-CoA yields 12 ATPs. Sources of acetyl-CoA. It is formed from pyruvate. It formed the beta oxidation of from it may be produced from beta oxidation of fatty acid. Important steps of acetyl-CoA. Oxalostate and acetyl-CoA is converted to citrate. This is the first step. Then isocitrate to alpha ketoglutarate. Then alpha ketoglutarate to succinyl CoA. Succinyl CoA to succinic acid. Succinate to fumaric acid. Then malate to oxalostate. Factors regulating TC cycles, enzyme citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase complex. Now, gluconeogenesis. I will discuss gluconeogenesis later. Thank you very much.